Hello my dear students, welcome back to the channel and thank you very much. You people are responding very well and you are benefiting from the channel. Today we are here to discuss another important topic. This is plant nutrition and this topic covers syllabus IGCSE, GCE O levels for Cambridge students and the students who are taking GCSE from Edexcel, AQA and OCR will also be benefited from this lecture today. Plant nutrition. Considering the nutrition, this is one of the important characteristics of living organism, means every living organism must feed on organic molecule. Now the source of organic molecule. Considering the source of organic molecule, we have divided living organisms into two categories. One, autotrophs or autotrophic organisms. These are the living organisms that create, that generate or that synthesize their own energy-rich organic molecules like carbohydrates, proteins and fats. For making these organic molecules, they collect raw material in the form of inorganic components from the environment and they collect the energy from the environment. Then these organic molecules they utilize in their life processes like respiration or growth. So examples of autotrophic organisms, a very well-known example are plants. So plants are autotrophs, means they can generate their own organic molecules during the process of photosynthesis and during this process they collect energy from the sunlight. The other category is heterotrophs or heterotrophic organisms. These are the organisms that do not synthesize their own organic molecules, or rather they collect prepared organic molecules from the environment and then utilize in their life processes like respiration or growth. And the well-known examples are animals. But our discussion today will be plants. So plants are autotrophic organisms. They make use of sunlight, collect energy from the sunlight, and they collect carbon dioxide and water from the environment and convert them into glucose and oxygen. This is the primary process of photosynthesis. For photosynthesis to take place in plants, so plants have got special organs, photosynthetic organs. These are the leaves. Within the leaves, there are special tissues known as mesophyll tissues. And these mesophyll tissues are we can simply call mesophyll. There are two types of mesophyll tissues, palisade mesophyll tissues and spongy mesophyll tissues. Their cells contain an organelle known as chloroplast. Within the chloroplast is present a pigment, a green color protein pigment that is chlorophyll. So this chloroplast is responsible for the process of photosynthesis and the chlorophyll present in the chloroplast is responsible for trapping light energy and converting it into chemical energy that is later stored in the form of glucose. You can see in this diagram that plants are collecting water and minerals from the soil in the water soluble form through their roots. These plants are collecting carbon dioxide from the air through the stomata of the leaves. Within the leaves, this carbon dioxide and water is converted to glucose and for this conversion energy is obtained from the sunlight. The process results the formation of glucose and oxygen. Oxygen diffuses out of the leaves during daytime as a waste product, whereas glucose is retained within the leaves, within the plant body, either respired as glucose, transported as sucrose, or stored as starch. Whereas minerals collected from the roots, these minerals are used to convert this glucose into other molecules like proteins, like nitrates are added up into glucose and glucose is reshaped up into amino acids and these amino acids later form proteins. Whereas magnesium is essential for making chlorophyll and chlorophyll is the most essential protein within the plant body because without magnesium there is no chlorophyll and without chlorophyll there is no photosynthetic activity. It's the chlorophyll that gives green color to the chloroplast, that gives green color to the mesophyll cells and that gives green color to the whole leaf. You must remember the equation for photosynthesis, whether it be the word equation or balanced chemical equation. during your exam, there could be a part that states write chemical equation for photosynthesis. Our examiner may ask you write word equation for photosynthesis. So for word equation, you can simply write in words carbon dioxide plus water, arrow, and as product you must write glucose and oxygen, and this must be in their names in words. And on the arrow, there must be conditions like sunlight or light and chlorophyll. Whereas when you are going to write about chemical equation, so you have to write symbolic equation like carbon dioxide as CO2 plus water as H2O, and then as product glucose as C6H12 and O6 plus oxygen as O2. But you need to balance this chemical equation, it is must process. So 6 carbon dioxide molecules react with 6 water molecules to raise 1 molecule of glucose and 6 molecules of oxygen. Or in terms of moles, you can say 6 moles of carbon dioxide react with 6 moles of water molecules, and as a result we obtain 1 mole of glucose and 6 moles of oxygen, and the conditions must be stated above the arrow or below the arrow like sunlight and chlorophyll respectively. So this you must know. Whatever is asked, you must respond accordingly. Photosynthesis is a process that occurs in 2 stages. One stage is known as light reaction, or this stage is known as light dependent stage of photosynthesis. During this stage, water molecule splits into its components like hydrogen and oxygen. So energy from the sunlight is actually required for this process. So that's why this process is known as photolysis. So what is photolysis? Photolysis is the breakdown of water molecule with the help of energy from the sunlight, and the water molecule is broken down into its components like hydrogen and oxygen gas. Oxygen gas escapes into the surrounding as a waste product, whereas hydrogen is used in the second stage. The second stage is known as dark reaction, or this stage is known as light independent reaction. In this stage, carbon dioxide is fixed with the hydrogen to form glucose. Hydrogen is released during the light dependent stage of photosynthesis and is used in fixing carbon dioxide in the dark reaction or in the light independent stage of the photosynthesis. So I repeat that photosynthesis, as we simply write in a single equation, it's not exactly a simple process. It is occurring in 2 stages. One stage is known as light dependent stage, in which water molecule splits into its components, hydrogen and oxygen, and energy of sunlight is used in this process. And chlorophyll is important in this reaction particularly. Whereas in the dark stage, hydrogen released during light reaction stage, it is consumed, it is used to fix carbon dioxide to form glucose during dark stage. During photosynthesis, what we have learned so far and what chemical equation states us, that we obtain only glucose, and you all know glucose is necessarily needed in respiration. But there are other molecules, other organic molecules required to raise different structures, to raise different functions in the plant cells. So where these molecules come from? Because photosynthesis only provides us with glucose. So where do we get the other molecules? It's an important question. Glucose produced during photosynthesis is converted into large range of other molecules for various plant needs, for various needs of plant cells. For example, this glucose is converted into cellulose molecules that later form cellulose fibers, and these cellulose fibers make cell wall of the plant cell. Similarly, some of the glucose is respired, and this glucose is respired to release energy, and this energy will be utilized in various life processes in plant cells like active transport, like protein synthesis, like other metabolic activities. Similarly, some of the glucose is converted into starch, and this is stored within the mesophyll cells. And this starch will be consumed later during dark times when there is no light, when there is no photosynthesis, when there is no glucose production at night. So this starch will be hydrolyzed to release glucose, and this glucose will be utilized during dark times, during night times for respiratory needs. Some of the glucose is converted into sucrose, and this sucrose is transported through phloem to different parts of the plant body, where this sucrose can be stored as starch in the storage organs, or this sucrose can be utilized for various other processes, like sucrose can be broken down to release glucose, and that will be utilized in respiration, particularly root cells or stem cells that do not produce any glucose because they do not carry any photosynthesis. This glucose is converted into ribose or deoxyribose sugars, and these molecules are used to make RNA nucleotides or DNA nucleotides respectively. DNA nucleotides, there is DNA that is an integral part of chromosomes and enclosed within the nucleus. This glucose can be converted into fatty acids or glycerol, and these fatty acids and glycerol can be used to make fat molecules, and these fat molecules can be used to make new cells, because every cell has got cell membrane and other membrane structures, and they are made up of fats. Similarly, these fats can be stored even in seeds, and these fats can also be used in other purposes like making other molecules for life needs. This glucose can be converted into amino acids by adding nitrates, by adding nitrogen from the nitrates, and these amino acids are raising proteins, and these proteins can be used as enzyme, can be used in raising new cells, and even these can be used for other life needed protein molecules. Chlorophyll is an important protein that needs magnesium. So if proteins are added with magnesium, so these can turn into chlorophyll, and these chlorophyll molecules raise chloroplast. So more chlorophyll, more chloroplast, more photosynthesis. So you can see that glucose that is produced during photosynthesis is converted into large range of other organic molecules, and this is important for growth, and this is important for maintaining healthy life activities. Now the question here arises that why glucose is converted into starch in cells, in mesophyll cells and in other cells. Glucose is an end product of photosynthesis, and this glucose is water soluble. It can be retained within the cytoplasm in some due concentration. If the concentration of glucose in the cytoplasm, particularly of mesophyll cells, it increases above that level, it will make the cytoplasm become more concentrated. It will reduce the water potential of the mesophyll cells, and it will affect the occurrence of other chemical activities. And it can also affect the ongoing process of photosynthesis, because if the product concentration rises and it makes an equilibrium with the concentration of the reactants, so process may stop. So in order to ensure that photosynthesis continues effectively throughout the day, so this product, that is glucose, must be carried out, must be carried away from the mesophyll cells, or must be converted into other molecules that do not affect the concentration of cytoplasm of mesophyll cells. Starch is insoluble, larger carbohydrate that stores large number of glucose molecules in it, and as being insoluble, it does not affect the concentration of cytoplasm. It does not affect the water potential of the cytoplasm of mesophyll cells. But during night, when there is no light, there is no photosynthesis taking place. So this starch is gradually hydrolyzed to release glucose that will be utilized in the process of respiration. Remember one thing, that respiration is all-time process. It occurs in every moment of every second. Even the plants are carrying photosynthesis, but they are respiring as well. But photosynthesis only occurs when there is light, when there is a daytime. So, in order to stay alive at night, when there is no light, plant need to carry on respiration, and this respiration is provided by the starch that was stored during the daytime. Now we discuss the structure of the leaf, because leaf is the photosynthetic organ. So it must be structurally modified to perform its job more efficiently. Like leaves are growing on a shoot, that shoot is known as leaf stalk. This leaf stalk is responsible to expose leaf well into the sunlight. So leaf is well elevated and well exposed to the sunlight, so it can absorb maximum of sunlight. Then we talk about leaf blade. So leaf blade is broad with the large surface area, so that more and more light it can absorb, and photosynthetic yield can be enhanced, can be increased at its maximum. Leaf blade is also known as lamina. When we talk about the thickness of the leaf, so thickness of the leaf depends on the number of tissue layers present inside the leaf. As we discussed earlier in the beginning, that carbon dioxide is the gas needed for photosynthesis, and oxygen is the gas that is produced during photosynthesis and is released as a product, as a waste product. So tissue layers, number of tissue layers ensure the effective gas exchange within the leaf. If number of tissue layers is more number of tissue layers are there, so leaf is thick, and exchange rate of these gases will slow down, because more layers, carbon dioxide to diffuse in and oxygen to diffuse out. So this will result the diffusion of these gases into or out of the leaf becoming a bit slow. And if the number of tissue layers are less, so leaf is thin and these gases can diffuse in or diffuse out, means carbon dioxide can diffuse into the mesophyll cells and oxygen can diffuse out of the mesophyll cells very effectively or efficiently. So when we study the cross section of the leaf, what we see, there is a topmost layer, a non-living cell based layer, that is known as cuticle. Cuticle is not comprising of any cell, remember. Cuticle is a wax, is a transparent waxy layer, and this cuticle is serving number of jobs. One, being waxy, being fat based, this is hydrophobic and it prevents water loss, direct water evaporation from the surface of the leaf. So uncontrolled water movement or water loss is prevented by the deposits of this cuticle. Cuticle being transparent. This cuticle refracts light. As you know, light refraction, when light moves from one transparent medium to another transparent medium, it bends. This is known as refraction. So this waxy transparent cuticle reflects sunlight and it ensures that more and more of the light enters the leaf inside. Then we have got underneath this cuticle, we have got a layer of epidermal cells. Generally as tissue they are known as epidermis, and it is the topmost layer, so we call it upper epidermis or upper epidermal cells. These cells are flattened in shape, compactly packed, and they are also transparent. These cells are producing cuticle and they are actually maintaining a particular thick layer of cuticle on the leaf epidermis. Second job they do perform, they give the upper boundary to the leaf, and the third job they perform is also refraction. Being transparent, they also refract light very well to the inside of the leaf. Remember one thing, upper epidermal cells do not contain chloroplast, so they do not carry photosynthesis. Right inside the leaf, underneath the upper epidermis, we have got palisade mesophyll layer, palisade mesophyll tissue. The cells of palisade mesophyll tissue are cylinder shaped, compactly packed, and they are highly enriched with the chloroplast. These palisade mesophyll cells, they receive light at maximum and at first instance. So they receive maximum light intensity, and in order to ensure maximum absorption of light and maximum rate of photosynthesis, maximum photosynthetic yield, so they do contain large amount of chloroplast in them. So maximum photosynthesis occurs in the palisade mesophyll, and these palisade mesophyll cells, you can see that these cells are provided with a very large sap vacuole inside that pushes the chloroplast near the cell membrane for effective absorption of sunlight, and their cell wall is also thin to ensure sunlight entering the cells very efficiently. Now, after the palisade mesophyll layer, we have a spongy mesophyll layer. These spongy mesophyll cells are irregular shaped and they are scattered, leaving spaces in between. These spaces are known as air spaces because within air spaces, air gets trapped, and this air is the source of gas exchange. The trapped air provides carbon dioxide to the mesophyll cells during photosynthesis and collect oxygen from the mesophyll cells that is released during photosynthesis. Spongy mesophyll cells do not receive maximum light intensity, rather they receive the sunlight that has been filtered out from the palisade mesophyll layer or that has been reflected from the soil and the tree trunk and has entered the leaf passing through the lower epidermis. So this light intensity is low. So they do not need much more chloroplast because they won't be able to carry much photosynthesis when compared to palisade mesophyll cells. So these spongy mesophyll cells do contain chloroplast but less in count when compared to palisade mesophyll cells. Now, air enters the leaf through the gaps that are present in the lower epidermis, and these gaps are known as stomata. But before discussing stomata, I must tell you that the cells of the lower epidermis, they are structurally the same as we have discussed about the cells of upper epidermis. Means they are also transparent, flattened, they also produce cuticle, and they also refract light to the inside of the leaf. But these cells, this lower epidermis, particularly in most of the plants, this lower epidermis has gaps in between. These gaps or pores are known as stomata, and through these stomata, air enters the leaf, gets trapped in the air spaces where gas exchange does take place. Carbon dioxide enters the mesophyll cells, oxygen leaves the mesophyll cells and enter the air spaces. Then this air, now rich in oxygen, diffuses out of the leaf through stomata, and a fresh air rich in carbon dioxide replaces it while entering through this stomata into the leaves. So these stomata are actually the gaps, are the areas through which air can enter or air can exit the leaf. Opening of stomata is controlled by specialized epidermal cells. These are known as guard cells, and in most of the plants, these are kidney-shaped or bean-shaped cells. These guard cells actually control the opening of stomata. Means they ensure that what particular diameter these stomata must be allowed to stay open or stay closed. They ensure that stomata are remaining open throughout the day in order to ensure a consistent and continuous supply of carbon dioxide. These guard cells are the only epidermal cells that contain chloroplast, and they can carry photosynthesis. They produce sugar during photosynthesis, and these sugars, they respire to generate energy in the form of ATP, and this energy is later used in fetching potassium ions inside these guard cells that will reduce the water potential of the guard cells, and in return these guard cells will fetch up or fetch in water from the surrounding, making them turgid, and they will let the stomata open. So in a way you can say this photosynthetic approach in guard cells will help these guard cells become turgid, and when they are turgid, they'll let the stomata open, and when they are more turgid, they'll let the stomata open at a wider diameter. But once these guard cells are flaccid, they are plasmolized, they shut down the stomata, they close the stomata. So no air exchange is possible that time. Within the leaves inside, you can see the vascular bundle consisting of xylem and phloem. Xylem is a tissue whose cells are responsible for continuous supply of water and the minerals in dissolved form from roots to the leaves, whereas phloem is responsible for transport of prepared food from the mesophyll cells in the form of sucrose and amino acids to the different parts of the plant body, particularly roots, stem, and the food storage organs. This was about the different components of the leaf and how they are specialized to perform their job in various perspective of photosynthesis. Compensation point. This is important to learn. Compensation point is the point or is the time when in the mesophyll cells or when in the leaves of the plant, rate of photosynthesis becomes equal to the rate of respiration. I told you earlier that photosynthesis and respiration, these are the two vital chemical processes that occur in plants. But for photosynthesis, light is required necessarily, and this process can only occur when there is light around, and in nature, the source of light is sunlight. So it can only occur during the daytime when there is a sunlight around. But respiration is the process that occurs 24 hours and it is not dependent on light. So every living cell in plant as well does respire, whether it's the daytime, whether it's the night time. The same is occurring within the mesophyll cells of the leaves. During daytime, they are carrying photosynthesis as well as they are carrying respiration. All carbon dioxide that is produced during respiration during the day, that is all consumed in photosynthesis. But as there is more light, more photosynthesis is taking place. So there is an additional demand of carbon dioxide. So that demand is fulfilled while collecting carbon dioxide from the air in the surrounding. So that's why during the daytime, plants are consuming carbon dioxide from the environment. But this photosynthesis results the formation of oxygen. Some of the oxygen is consumed during respiration, that is occurring inside the cells of the leaves, and excess oxygen is released into the surrounding. So that's why during daytime, during light time, plants do not collect oxygen from the air, rather release excess oxygen into the air, but collect carbon dioxide, that is the photosynthetic need. At night time, when there is no light around, so plants cannot carry photosynthesis. But in order to stay alive, they need to respire. For respiration, they need oxygen. But this oxygen now they have to collect from the air around the plant. So at night, in the times of darkness, plant collect oxygen from the air in their surrounding, and as a result, they release carbon dioxide into the surrounding, because at night when they are not carrying photosynthesis, so this carbon dioxide cannot be consumed, rather it is released into the surrounding. So I repeat, at night plants cannot carry photosynthesis, but they do carry respiration. For respiration, they collect oxygen from the air around and they release carbon dioxide in return into the surrounding. But when it's the sunrise time, when sun is there but it is with the low light intensity, or at the time when the sun is about to set, So there is low light intensity. So at low light intensity, what we call dim light, rate of photosynthesis and rate of respiration both become equal, because during respiration, carbon dioxide produced all is consumed during photosynthesis, but there is no additional demand for carbon dioxide because light intensity is very low. So carbon dioxide that is produced during respiration is all consumed in photosynthesis, and oxygen produced in photosynthesis is sufficient enough to compensate for respiration. So there is no additional oxygen left that could be released into the surrounding. During dim light states, when it's sunrise or when it's sunset, so plants do not manage any gas exchange with the air around. They do not collect any carbon dioxide or any oxygen from the surrounding, and they do not give off any carbon dioxide or any oxygen into the surrounding. So these particular times of low light intensity or dim light, this is what we call compensation point, where the respiration and photosynthesis, they are meeting the demands of each other. So no gas exchange with the surrounding air. This is compensation point. And now we need to discuss some of the factors that can affect the rate of photosynthesis. These factors, when they fluctuate, when they increase or decrease in their values, they can also influence the rate of photosynthesis of the plant accordingly. These factors include light intensity, carbon dioxide concentration, and temperature. First of all, we will discuss about light intensity. As you know that the light is the source of energy for the process of photosynthesis. So it has been observed that light intensity has got its direct impact on the rate of photosynthesis. If the light intensity increases, rate of photosynthesis also increases. And if you just consider this curve on your screen, you can see that initial increase in the light intensity will result a very steep rise in the rate of photosynthesis. But later you can see the rate of photosynthesis is rising, but the curve is bending, means the rise rate is slowing down. And afterwards, the rate of photosynthesis have become leveled off, means it has become constant, even though the light intensity has increased. Why this is so? Remember one thing, there is a concept of limiting factor. Limiting factor can be the reaction component or can be the environmental component in which reaction or photosynthesis is taking place. Limiting factor is always in its limited approach or limited supply. If it is a reaction component, so it would be in the limited supply throughout the course of chemical process or throughout the course of photosynthesis, as we are discussing here photosynthesis. Like carbon dioxide could be in the limited supply, or temperature being the environment, being the component of environment, could be in the optimum approach or could be in the fixed approach. So this temperature or carbon dioxide or light intensity on their turn can serve as limiting factors due to their limited approach or limited supply. When we are carrying out any experiment to study the effect of any of the components of photosynthesis, any of the components in the environment where the photosynthesis is taking place, so we are going to study one component and the other components, they are kept fixed or at their optimum values. Okay, so this would help us to study the effect of only fluctuating factor, not the others. So in this case, in the case of light intensity, when it is increasing, in the initial stages, light itself is serving as limiting factor. In the initial part of the curve, you can see in A. So light intensity, even though it is increasing, but itself it is serving as limiting factor, because all the hydrogen produced in photolysis is being consumed in fixing carbon dioxide. But later light intensity has increased. It can release more hydrogen because it can carry more photolysis. but limited supply of carbon dioxide has limited the rate of reaction to this constant value. Okay, so here we have studied when the curve is rising, light intensity is itself a limiting factor. When the curve has become constant, temperature that is fixed, or carbon dioxide that is in the fixed value, fixed supply, that has turned out to be the limiting factor. When we study the effect of carbon dioxide, we see that increasing the carbon dioxide concentration is also increasing the rate of photosynthesis, and this is also a direct impact, or you can say directly proportional. But you can see initial increase in the carbon dioxide concentration, there is a very sharp rise in the rate of photosynthesis. But after some time, this rise rate slows down and later it becomes constant. Even the carbon dioxide concentration is increasing. And this is because of the limiting factors operating here. Initially, carbon dioxide itself is the limiting factor where the curve is sharply rising. But later, fixed temperature or light intensity are serving as limiting factors. So they do not allow the rate of photosynthesis increasing above this level. Remember one thing, when the curve has become constant, it does not mean that reaction has stopped. It means reaction is occurring, but it cannot occur beyond this rate, beyond this space. The third important factor is temperature. As you know, that photosynthesis is also a chemical reaction, and this photosynthesis is also catalyzed, controlled by enzymes. So increase in temperature will increase enzyme activity. We have observed that initially there is a direct impact on the rate of photosynthesis. As temperature increases, rate of photosynthesis also increases because enzymes are becoming more and more active. Kinetic energy of the enzymes and substrate molecule increases, so reaction rate goes high. At optimum temperature, rate of photosynthesis reaches maximum. But if the temperature is increased above optimum, rate of photosynthesis drops. Why? Because above optimum temperature, enzymes start getting denatured and their active count starts decreasing. So reaction rate starts decreasing. So photosynthesis starts decreasing. And if temperature continues to increase, so at particular temperature, all the enzymes get denatured. So photosynthesis eventually stops. So you can say initial increase in the temperature till optimum, it helps in increase in the rate of photosynthesis. But as temperature increases above optimum, it denatures the enzymes and photosynthesis drops. When all the enzymes are denatured, photosynthesis completely stops. Now experimentally, to consider the effect of these factors on the plants, really they do affect the photosynthesis or not. Before introducing any plant to such conditions for experimenting photosynthesis, we need to destarch them. Why it is important? Because when we introduce a plant to the photosynthetic conditions, it does carry photosynthesis, and photosynthesis produces glucose and oxygen. Oxygen being transparent gas, it escapes, while glucose is converted to starch. And presence of starch can confirm us that photosynthesis has taken place or not. For this, we conduct iodine solution test. But during the course of experiment, whether the starch was produced or starch reflecting at the end of experiment was the one that was already present before conducting experiment. So this raises question mark over our experiment, over our results. So in order to reduce any errors, any ambiguity, what we do? We collect potted plants for photosynthetic experiments. Why potted plants? Because potted plants can be easily introduced to the desired experimental conditions, and studying growth rate becomes easy over the potted plants. experimental study can help us understand the effect of these factors, that how do they influence the rate of photosynthesis when fluctuate. But before going into experiments, we need to select plants, and we prefer to select potted plants because it becomes easy for us to introduce these potted plants into the desired experimental conditions. And these potted plants must be de-starched first, because every photosynthetic experiment we need to confirm the presence or absence of starch. That is the ultimate end product of photosynthesis, because glucose produced is immediately converted to starch within the leaves, whereas oxygen escapes being transparent gas. So in order to confirm photosynthesis has taken place or not, and particularly when we are talking about the land plants, so we confirm the presence or absence of starch. But in order to ensure that every starch molecule that reflects in the leaves is produced during the course of experiment, we need to de-starch the plant first before introducing it into the photosynthetic experiment. If we do not do so, we will not be sure about that starch reflecting is the one produced during photosynthesis during experiment, or is the one that was already present in the leaves before conducting experiment. So in order to avoid this confusion, this ambiguity, we de-starch plants before introducing them into the experiments. How we can conduct de-starching? We will place the selected potted plants for 3 to 5 days in the dark room. During dark time, no photosynthesis as there is no light, but in order to stay alive, plants will carry respiration. All the starch stored will be gradually converted to glucose, and that would be consumed in respiration. So within this dark time period, the plants will consume all their stored starch and their leaves will become starch free. Now these potted plants can be introduced into the photosynthetic experiments with the desired conditions. Once the experiment time is over, so leaves are collected from the experimented plants and they are processed for the presence or absence of starch, and we conduct starch test with a bit different procedure. First of all, the leaves collected from the experimented plant, these leaves are placed in the boiling water for about 20 to 25 minutes. This boiling treatment of these leaves results cells being dead, cytoplasm being dead, enzyme get denatured, and this prevents any other chemical reaction occurring, and permeability of the membranes, particularly the membranes of chloroplast and the cell membrane, it increases. After this boiling treatment, leaves are placed in the hot ethanol for about 10 minutes. Ethanol must be pure, that what we call absolute, or must be 90%, not diluted than this. Hot ethanol will serve as a solvent and it will absorb all the chlorophyll from the mesophyll cells in the leaves. And this hot ethanol, as it has got low water potential, so it will also absorb all the water from the leaf. So this will turn the leaf becoming brittle white, yellowish white leaf, and these leaves will lose their water, will lose their flexibility, and their chlorophyll is also lost to the ethanol, and ethanol itself will turn green. After the ethanol treatment, we place the leaves in warm water for 5 minutes. During this time, water enters the leaves, enter the leaf cells, and they restore their flexibility and elasticity. Then we take out the leaves from the water, we place them, stretch them on the white tile, and pour a few drops of iodine solution. If the leaf or any part of the leaf has carried photosynthesis, it would contain starch and it would turn blue-black. And if there is no starch in the whole leaf or in any part of the leaf, so that particular area or the whole leaf will turn yellow-brown, that is the color of the iodine solution. So this is how we conduct starch test after the experiment is over. Before going for experimental discussion of these factors, we must study the effect of minerals on the plant growth, effect of nitrates and effect of magnesium on the plant growth. Nitrates are very essential because nitrates are the source of nitrogen, and this nitrogen is incorporated into glucose when they are converted into amino acids. And these amino acids later they form polypeptide chains, and these polypeptide chains actually make proteins. And these proteins are essential for plant growth, means more proteins, more enzymes, more chemical reactions, more photosynthesis, more chlorophyll, so there is a healthy plant growth. Similarly, magnesium is also another important mineral that is essentially required for chlorophyll. Without magnesium, there is no concept of chlorophyll. So, if there is more magnesium, more chlorophyll is being produced, and more chlorophyll means rate of photosynthesis will occur at an increased rate, more food is being produced. So again we see that there is a high growth rate. So we can conduct simple experiment to study the effect of these minerals. Like we can take two plants and we can provide them, one can be provided with nitrate rich soil and other can be provided with nitrate deficient soil. So we can see that the plant with the nitrate rich soil will show a higher growth rate in few days later, where the plant with the no nitrates in the soil will show stunted or reduced growth. Okay. Similarly, plants provided with high magnesium contents in their soil will grow healthy, taller with more number of leaves, whereas the same species plants grown on a soil with no magnesium at all, so they will be having a very reduced growth, leaves turning yellow because no magnesium, no chlorophyll. So the experiment on your screen, this is also a modified form of it. The same species plant has been taken. Okay, the one that is provided with the nitrates and with the magnesium, and the same species plant provided with soil without nitrates, and the same species plant provided with the soil with no magnesium. And you can see the plant with magnesium and nitrate that is growing healthy with more height, with more number of leaves, whereas plant who are deficient of either nitrates or either magnesium, so they are having weak growth and yellow leaves, particularly with the magnesium deficient plant. And now we discuss the factors. Experiments we are going to discuss now. In the very first experiment, we will study whether light is necessary for photosynthesis or not. In this case, we will select a healthy potted plant and we will destarch it first using the same procedure as I told you people earlier. After destarching is completed, we will take out the plant, we will select some of its leaves and we will place stencils on them. These stencils are actually dark papers with some words or some shapes cut inside, and they are pasted on the leaves. Now these plants will be introduced into bright sunlight. They will absorb carbon dioxide from the air and they will receive light. After the photosynthetic duration, the experiment duration, let's suppose it's 2 hours or 4 hours, we collect the leaves, pluck the leaves with the stencils and we conduct starch tests. Now we see that parts of the leaves, they were receiving light. They did color blue-black, and parts of the leaves, because of stencils that could not receive any light, they were stained yellow-brown. Why? Because parts of the leaves that received sunlight, they carried photosynthesis and they produced starch. Whereas the other parts of the leaves that did not receive light because of dark paper around, they were consuming carbon dioxide from the surrounding, but they were not receiving any light, so they did not carry any photosynthesis, so no starch reflected in their cells. So that's why these parts were stained yellow-brown. In the second experiment, we are going to study the effect of carbon dioxide. Okay, how we can study? We can take two plants of the same species, same size, and we place two plants in the transparent jars. Why transparent jars? Means these plants will be receiving sunlight, but they would be partially isolated from the air in the atmosphere. In one jar, we add, in both of the jars we introduce funnel. In one jar, jar A, the funnel is added with the sodium hydroxide, and inside the jar along with the plant we have introduced potassium hydroxide along with the plant. Okay. In the other jar, we did not place potassium hydroxide and we did not place any sodium hydroxide. We let the jar receiving air from the surrounding, and that air is supplied to the plant. But remember one thing, both of these plants should be of the same species, same size, and they should be destarched first before introducing into the experiment. We'll place the two plants with their jars intact into the open light, into the open air, and wait for a few hours. Let's suppose for 4 hours. After 4 hours, we collect leaves from the plant in jar A and conduct starch test, and we find they are turning yellow-brown. Whereas when we conduct starch test on the leaves collected from the jar B, they were stained blue-black. Why is it so? Because sodium hydroxide was absorbing any carbon dioxide that was present in the air before it entered the jar. And within the jar, potassium hydroxide also ensured that no carbon dioxide should be there in the air inside the jar, because both sodium hydroxide and potassium hydroxide are carbon dioxide absorbent. And you can see in the experiment design that even plant pot soil has been covered with the polythene bag, so that any carbon dioxide produced by the microbes working in the soil, that should not be introduced into the air. So leaves of plant A did not receive any carbon dioxide even though they received light. So they did not carry any photosynthesis, no starch in their leaves. So that's why these leaves turned yellow-brown. Whereas the leaves of plant B are leaves of the plant placed in jar B. They received light as well as they also received carbon dioxide from the air that is entering the jar. So they carried photosynthesis, produced glucose that later turned into starch, and that was confirmed by turning the leaves blue-black with the iodine solution. So that confirms that carbon dioxide is important for the process of photosynthesis. The third experiment we can conduct to confirm that chlorophyll is important for photosynthesis, and it's actually the chlorophyll that is responsible for photosynthesis. So what do we do here? We collect special plants that are known as variegated plants. These variegated plants are the ones, their leaves have chloroplast in patches. Areas of the leaves that contain chloroplast, chlorophyll, they are colored green, and rest of the parts of the leaves, they are colored yellow-white. So we collect variegated plants with the healthy leaves and we destarch first, and after that we allow these variegated plants to carry photosynthesis in open sunlight for 2 to 3 hours. Once the experiment duration is over, we'll pluck a couple of leaves and we'll conduct starch tests. And what we observe, that parts of the leaves that were green before conducting starch test have now turned blue-black, and parts of the leaves they were yellow-white before the conduction of starch test, they have now turned yellow-brown. And what this reflects? This reflects that parts of the green parts of the leaves were containing chloroplast, chlorophyll. So photosynthesis did take place there, and there was starch now. And the yellow-white part of the leaves did not contain chloroplast, chlorophyll, so they did not carry photosynthesis. So no starch there. So that's why they turned yellow-brown. Now at the end, we are going to discuss the effect of fluctuating light intensity. Means when light intensity is low, rate of photosynthesis is low. When light intensity is high, rate of photosynthesis is high. So in order to confirm it experimentally, we'll conduct a study. During this experiment study, we'll use a water plant. And this water plant would be either hydrilla, Canadian weed, duckweed, or pondweed. So these are water plants and that grow inside the water bodies. Why we are discussing this experiment with reference to these water plants? Because we need to compare the rates of photosynthesis, and this comparison is only possible with the help of water plants, because now we will not be going for starch test. We'll study the oxygen production, the volume of oxygen produced during photosynthesis that can be measured and later compared with different light intensities, or number of bubbles of oxygen produced per unit time or per minute would be discussed. Okay, so how we are going to have this experiment? We'll have a lamp as a source of light. We will have a water plant well inserted into a hydrogen carbonate solution. Hydrogen carbonate solution will be a source of carbon dioxide, because hydrogen carbonate solution will provide carbon dioxide to the water plant. Okay, we'll keep this water plant in the hydrogen carbonate solution at a distance from the light source, and between the light source, that is the lamp, and the water plant, we will place a thermal filter. This thermal filter is transparent. It will only allow light to reach the plant, and any heat intensity in the light, it will be absorbed by this thermal filter. Now we will fluctuate light. We will change the light intensity, and this could be done by three means. For example, if we change the distance between plant and the lamp, light intensity can fluctuate. We'll bring the lamp closer to plant, light intensity will increase. We'll bring lamp away from the plant, light intensity will decrease. But when we are doing so, the power of the lamp and the color of the light should be kept constant. The second mean is we will change the power of the lamp. But when we are changing the power of the lamp, we'll keep the distance between the plant and the lamp fixed. That should not change. So we can have different powers, low power or high power lamps, bulbs can be used. So that will actually affect the power of the lamp, and changing the power of the lamp will change the intensity of the light, and it will affect the rate of photosynthesis in the water plant. And the third mean is by changing the light color. As you know that white light is consisting of different light color. So we can take three basic colors like red, blue and green. Blue color has got the least light intensity, whereas red color has got the highest light intensity in the visible light spectrum, and green color is somewhat in the middle, but green light is least absorbed. Okay, but when we are using color filters or different colored lights, we should keep the power of the lamp fixed, and we should keep the distance between the lamp and the light source fixed. How we can measure the rate of photosynthesis, at each light intensity, we can count the number of bubbles produced per minute that are rising to the gas jar above, because when oxygen will be produced, it will be released in the form of bubbles. So these bubbles can be counted rising to the gas jar, or we can collect the gas in the gas jar, and later we can confirm that the gas is oxygen by introducing a burning splint into it, or we can also compare the volumes of gas collected at different light intensities. So we can see at higher light intensity, when rate of photosynthesis is high, more oxygen gas in more volume is collected in the gas jar compared to the gas collected at low light intensity. So during this topic we have discussed today, we have discussed about photosynthesis, about plant being autotroph and being photosynthetic in nature. We have also discussed photosynthesis that is occurring in two stages, in the light dependent stage and the light independent stage. We have also discussed that glucose is produced primarily in the photosynthesis, and that glucose is later converted into large range of other organic molecules as needed by the plant. We also discussed about different factors like light, temperature, and carbon dioxide concentration, how they affect the rate of photosynthesis, and we have also discussed experiments in this context. And we have also discussed the limiting factor, and we have also discussed effect of minerals in the plant growth. So that was all for today. Stay tuned for further updates, for further lectures. Thank you very much.